Hi, everyone. Uh, I'm glad to be here. This is my first time here. So, whatever the culture is, I guess I'm setting it today. So, I was told that I fit in culture-wise. So, I have the same font as Kirk did. So, that's a good thing. Um, my name is Ben Langmead. I'm an assistant professor of computer science from Johns Hopkins. And today I have a presentation that, in the spirit of us being here all week, I'm going to not deliver any sort of talking points about my lab or give you a broad overview of what we do. If you would like, you can go to my lab website, which is here, or my Twitter feed, and figure out about that, or talk to me because I'm here all week. Instead, what I'm going to try to do today, so today I might occasionally refer to it as a talk, but really this is a tutorial. The way I've structured the tutorial is that I have a few exercises that I'll launch into in the middle. 45 minutes is not a lot of time to do a sort of workshop set of exercises. So in the spirit of great cooking shows, I've pre-prepared a great deal of it. I've downloaded some data. I've got some cells in my R Markdown notebooks that I've already run, so that when it comes time to do the demonstrations, I'm mostly going to talk through what the code is doing and then run some of it. But the idea is I also have this repo where all the code is located. So you can go there and you can redo them later if you like, edit them, correct them, things like that. And I also have the slides themselves up on SpeakerDeck already in case you want to follow any of the links from the slides. Okay. And I also have the related readings. They didn't show up in the schedule, so I thought I'd put them in the slides so that if you do go look at the slides, you can simply click the links to get to the related readings. Okay, so onto the content of today's tutorial. So the tutorial today is really about trying to use the huge amount of archived public sequencing data that we now have in order to answer scientific questions, your scientific questions or your collaborators' scientific questions. And this is not something we're necessarily trained very much in doing, but I think it is something that people do increasingly. And as computationalists, which many of us are, I think this is a really important topic to tackle. So the sequencing revolution certainly has the potential to sort of empower everybody to ask and answer big questions about sequencing data, nucleic acids, and other things that we can assay with sequencing data. But there are barriers to doing this, right? Real practical barriers, many of them computational barriers, which means that the fruits of the sequencing revolution have, in practice, not been totally democratically shared. You know, it's accrued mostly to groups that can figure out the computational part. One thing that computationalists can do is to try to take the enormous amount of raw data that's available in archives and imbue them with more queryability. You know, make them a little bit easier for everybody to use. So that folks who work in labs that don't necessarily look like this, maybe folks who work in labs with less resources and who don't necessarily have a lot of computational savvy, can get a lot of value out of that data as well. So let's talk a little bit about the public data that's available now. So I need to be able to convince you that these are useful resources. So I need to convince you that there's a lot of it. And I need to convince you that it's valuable, that it might be something you might want to use on a day-to-day basis. So let me start with a plot. This is not the usual how much faster our sequencer is getting or how much cheaper our sequencer is getting plot, but it's another intimidating plot. This is a plot about how much data is in sort of the premier archive that we have for archiving sequencing data sets called the Sequence Read Archive. Raise your hand if you've downloaded data from the Sequence Read Archive. Okay, maybe a third, maybe a half of you. So this is not the only place where raw sequencing data is stored, but it's probably the biggest one. And it has a lot of data in it, in part because the culture of the field involves journals often forcing authors to upload their raw data to archives before they can publish their papers. And the rate at which it's been growing is shown here. That vertical axis is on a log scale, so it's a little bit difficult to interpret. But let me highlight two recent doublings. of the size of the sequence read archive. So those two intervals you see up there, the blue and the red intervals, show 2 times where the sequence read archive doubled in size from 4 to 8 and then 8 to 16 petabases. It's somewhere around 20 petabases right now. And you can see that the doubling time interval is in one case 12 months, in the other case 18 months. 12 months is unusual. You might see there's kind of like an elbow in that blue line right around the end of that 12-month interval. That's from one project. That's from the TopMed project, deposited a ton of data in October 2016. So that's kind of an anomaly. In general, the rate of growth, though, pretty much tracks with what you might call a Moore's law rate of growth, doubling every 18 to 24 months or so. So it's getting really big. Now, not every piece of data in the sequence read, those of you who raised your hands might know this already, not every piece of data in the sequence read archive is perfect and pristine or annotated with relevant or even correct metadata. All right, so there's a lot of issues with getting good data out of the sequence read archive. But there's also no question that there is a ton of valuable data in the sequence read archive, data that to us would be beyond our ability to collect, or it would come at a huge opportunity cost for us to collect. So things like data from rare phenotypes, data from difficult to obtain tissues, highly purified tissues, ancient DNA. There's a long list of examples, and in your own field, you probably can think of better examples than the ones that I just gave of data sets that are valuable. Some of them are also just quite large, right? They're valuable simply because of sample sizes. And as bioinformaticians, maybe there's also something empowering about the existence of all this archived data. Like, what if we have hypotheses? We do, right? Sometimes. We have the ability sometimes to test scientific hypotheses without having to generate a lick of data, which can be hugely valuable, right? Because otherwise, if you have to, from scratch, get the team of collaborators together in order to generate data and pay for that data, that obviously is often going to be the bottleneck. It's actually really hard to get from scratch, from hypothesis to generated data, if you are not yourself equipped to do that. It could also mean that bioinformaticians can, in the process of designing and executing a study, get involved earlier, because if we know how to marshal the public data, we can often say something about how these hypotheses look purely with respect to public data, or what the study design should be by using public data. So I think for bioinformaticians, there's a self-empowering aspect to being able to use public data more readily. Now, public data is not that easy to use, right? Public data in the sequence read archive is not queryable per se, right? It's a lot of big compressed blobs of raw data. There's like one big blob of a file for every run accession that's stored in the sequence read archive, basically for every sample that's in the sequence read archive. So it's like, I like to use the analogy, it's like the early days of the World Wide Web. There was a World Wide Web before there were search engines, believe it or not, and it was hard to use. And search engines came along and they completely changed the way people use the web. And now we're used to being able to put a query into a search box and literally less than a second get responses. So the challenge of being able to make this data more usable consists of a couple of things. Certainly, much like the web search engines, it includes indexing. And so I like this quote from George Bernard Shaw, who said, "An index is a great leveler." Right? You might have people who know the entire contents of the book, but that's not quite so important if the book has a good index, and now anyone can go look up the appropriate contents of the book. But indexing is not the only technique we have. We can also summarize. So we can condense and summarize, remove irrelevant data, and just otherwise try to sample even, sometimes just to try to shrink the size of the data. So even a summary would be an improvement, did not say Bernard Shaw. I said that. But so to make sequencing data easier to use, we're going to use some combination of indexing and summarizing. And computer scientists love indexing. We've studied it for a long time. Statisticians love summarizing. Computer scientists love summarizing. These are great things for computationalists to think about. One thing I won't talk about, but which is a great topic that's been written about, including by at least one other speaker today, is how to index sequencing data raw, just in its raw form, and just try to make the raw data easier to query. a little bit like in the spirit of Google, right? Google makes the raw web pages to some degree. They're a little bit summarized, but basically Google makes raw web pages queryable. So why can't we make raw sequencing data queryable? Well, you can, and there's really great work in what data structures you might use to do that. They include things like Bloom filters, Bloom sequence trees, quotient filters, FM indexes, and things like that. This is not my emphasis today. Everything that I show you today will involve some indexing. I'm sorry, some summarization prior to indexing. So these are great references. Reading these papers is very fun as a computationalist. I'm going to talk about techniques that summarize more than these techniques do. Okay. I'll talk about a few efforts and show you exercises related to a few efforts that I've been involved in, but there are many efforts to summarize public sequencing data. That box there is from one of the papers on the reading list, a review paper about cloud computing and summarizing public sequencing data. Those are mostly, I think all, RNA-seq examples, RNA sequencing examples, but down here I added a few more that are not necessarily RNA sequencing. It would have been even better if I'd added a metagenomics one, like Experiment Hub is a good example of a metagenomics one that I failed to add. But there are many examples of public repositories of summarized archived sequencing data. Okay, I'm going to talk about basically 3 different layers of summarization and ways that you can take advantage of them. And I'm going to show examples where you can take advantage of data at each of these 3 levels. These are 3 different projects that came from my lab and from collaborators at Johns Hopkins, including especially Jeff Leake and his lab, but also others like Andrew Jaffe and Casper Hansen. And I'm going to take these one layer at a time, and I'm going to show you basically how the data that they generate can be used to answer a question. I'm going to get started by showing how data that comes from this tool called RailRNA, which is a spliced aligner, and I'll explain that in a moment, you can start to mine the data from that sort of tool to ask and answer questions about splicing. A quick word on that. So really the only background you need to understand most of what I'm going to talk about in this talk is exon-exon spliced junctions. All right, so the data that we've summarized from public archives is mostly human RNA sequencing data. So we're sequencing messenger RNAs derived from humans. They are mature messenger RNAs, so the introns have already been removed. This means that when we go to solve the alignment problem, the very first problem we usually have to tackle when we analyze the sequencing data, there's an extra wrinkle to the usual alignment problem. We don't just have to figure out where the reads came from with respect to a reference, but we have to respect the fact that sometimes they overlapped an intron that was spliced out. If they overlapped an intron that was spliced out, that means that their point of origin is actually in pieces, right? Because it's split up across either side of the splice junctions, either side of the introns. So a great thing, so the algorithm has to deal with this, and that's what makes a spliced aligner a little bit more complicated than a typical read aligner. But it also gives us this wonderful thing that we can summarize, which is how often do we see a given splice junction in a sample? And that's the main thing that we're going to be talking about querying and summarizing. Okay, so in this picture, that wormy thing up there is a messenger RNA, a pre-mRNA. It has not yet been spliced. The black bits are the introns, which are going to be spliced out to make that continuous orange tubey thing. So spliced alignment has to figure out how to align the reads, including those ones highlighted red that go across the splice junctions. Okay? So we put together a suite of software that was very, very scalable because we wanted to run it against lots of public data. How we did that is not really a subject that I'll go into today. It would take too much time. Happy to talk to you about it elsewhere. We use things like Amazon Web Services and Elastic MapReduce and cloud computing. But suffice it to say that at the end of the day, what we get is a summary, which is basically like a big matrix, where the rows of the matrix are all splice junctions, so intervals of the genome that were spliced out, and the columns are samples. So for every individual we studied, we can count how many pieces of data, how many reads supported that splice junction in that sample, how many reads seem to go across that splice junction in that sample. So more reads means more often, we saw it more often. right? So it's going to be proportional both to how often that gene, how highly expressed that gene is, and to how frequently that splice junction occurs in that gene. Okay? And so we compile all that into a matrix that looks like that. Rows are junctions, columns are samples, the elements are just counts for how many times we observe it. And so now, let me pose a question and then I'll switch over to an R markdown notebook and show you a little bit about how we can answer it. So the broad question we want to ask here, we have these things called gene annotations. Many of you have probably worked with gene annotations, but it's curated collections of genes and their isoforms so that people can sort of know ahead of time what are the possibilities for what this gene might look like when it's expressed and then spliced. It's not too surprising for me to say that these are not complete, right? So these gene annotations are some of what we see in nature. Question that we wanted to ask is, since we've analyzed a lot of data, maybe we can start to look at, well, what fraction of what we found was annotated versus what fraction of what we found was not annotated. That'll give us some idea of a measure of completeness for the gene annotation. All right, so at this point, let me switch to the cooking show and go to our studio. And what I have here is a notebook where we're going to basically open that matrix that I just showed you, do a few very quick summary statistics over it, and then pose this question about what fraction of the junctions are annotated. So I've pre-run several things in this notebook. I'll just scroll down and say quickly what they are. This is loading some libraries in R. This is setting a certain directory. These are a couple of functions that are going to help us read the files because they're in a particular format. And then this one I definitely had to run ahead of time because that's the one that actually parses the great big matrix with all the splicing data. Okay, so that I've run already. But what I can run for you here is now I'm going to parse the metadata. That's not so big, so that I can do right away. Now I want to print a couple of summaries over that big matrix of splicing data. I just want to show you how many rows and columns it has. So the number of junctions is the number of rows, that's 81 million. The number of columns is the number of samples, that's about 50,000. So we analyzed about 50,000 human RNA-seq samples. So this matrix has 50,000 columns. Okay. Now I want to show you one way that we can pose that question that I mentioned before. I'm going to make a certain kind of plot that shows basically as we become more and more strict about what we think is a true junction, we're going to ask what fraction of those or what number of those are in gene annotation. So let me first of all, before I do that, let me just show you really quick what some of the data in this matrix looks like. Here it is. This is the big one. Let me just do, this is going to be a small font. Wow, it's a big screen, so it's not that bad. These are just the first 6 rows in that matrix. So each of these is a junction. It's telling you the length of the junction, in other words, how much sequence was spliced out. And it's telling you a couple of summary statistics for how often that junction was observed, one of them being in how many different samples was it observed. Now we have 50,000 samples. And so for these junctions you see here, they only occurred in a handful. And that's actually pretty common. And for the most part, those are probably wrong. Those are probably mistakes made by the computational spliced aligner. We don't necessarily trust them if they only occur in a few. But that's not necessarily the case. It could be something more like splicing that occurs, but it occurs very randomly. And that splicing might, in turn, not be very important if it only occurs very rarely and randomly. We don't quite know, which is why we keep the rows in the matrix. We don't throw them away. But suffice it to say, a lot of the rows in this matrix is going to be junctions for which there's actually not much support. All right, so having said that, let's actually make a plot. Okay? Yeah. Yeah. It could, well, so that summary, let's go back. So this column is the number of samples for which it had non-zero count, and then this is the total count. So you can see the count is also often quite small. Right? So for these examples you see here, it probably, like, they're equal in all but that case, right? So in that case, some of them had 2 or 3. Okay, so now let me make a plot that starts to get at this question of what fraction are annotated. I've got a plot where the horizontal axis is basically a filter. I'm putting a more and more stringent filter on the junctions that I'm willing to consider. As we go to the right, we're requiring that the junctions occur in more and more of the samples. Right? The more samples they occur in, the more confident we can be that it seems to be a real phenomenon that's repeatable and occurring across different individuals. The vertical axis is the count, and I've got counts for 4 different kinds of things. Junctions that are totally annotated, which is red, which you'll see as most of them. Junctions that have annotation for both ends, but those 2 ends aren't ever on either side of an intron in the annotation, so that I call an exon skip. Then I've got purple, which is that one or the other end of the splice junction is annotated, but not both. In other words, you see a junction beginning or ending like that in the database, but never beginning and ending like that in the database. And then neither is that blue one. And it's cumulative, so that's why it's going sort of like up like that. And what you see is, not surprisingly, the numbers go down as we get to the right, because our filter is getting more stringent. And then furthermore, as we get farther to the right, the lines get closer together. That's perhaps also not surprising, because if it's more and more repeatable and occurs in more samples, it's more likely to have been curated and more likely to occur in the annotation. But there's this bit in the middle where the lines are kind of spread out. And we wonder, what are these differences? Are these big differences or what? So I have another quick summary I can show you, which is a table where I've picked a few, basically almost like cut that previous plot at a few points vertically, so that I can tell you for a particular requirement, percent of samples, like requiring, for example, that the junction occur in at least 10%, in other words, at least 5,000 or so of these 50,000 samples. If we have that requirement, what fraction of them are totally new, where neither end of that junction occurs in particular in the gen code gene annotation in this case? And the answer is somewhere around 13%. Right? So we can make tables like this. We can ask ourselves whether we're surprised by these numbers. We can ask ourselves whether these are what we expected. Certainly, I think what it hammers home is something that folks believe but don't necessarily always assume when they do their analysis, which is gene annotations are not complete. Right? There's a great deal of splicing that could even be called common, right, occurring more than 5% of samples in the archive, but that aren't included in gene annotations. So that is exercise number 1. Allow me to switch back. So that was an analysis at, yeah, question. Yes, the 2 coordinates in the strand. So that was an analysis at the level of, what if we have a giant matrix with all the samples as being columns of that matrix? But another way we could try to analyze data is to zoom in on studies, and that's what the recount project is about. This is another way that we've summarized the data, more on the level of individual studies. So recount was published last year, and if you go to the recount website, it's got an interface that my colleague student Leo Collado Torres created that uses Shiny, and you can do things like put a search term in and get back only the studies that have an abstract or some other metadata that match that search term. So here I've put in T2D for type 2 diabetes. I get back some abstracts that have T2D for type 2 diabetes. You can put in whatever search term you want. But once you've zoomed in on the studies you're interested in, it gives you kind of like this bevy of links over here to all the various different ways that we've summarized those studies so that you can load those summaries in and start working with them quickly. And we've summarized basically at 4 different levels. The level of junctions is what I just showed you, so that's not surprising. So we can easily, if we can do it for the whole compilation, we can certainly do it for the studies. Exons, individual exons in the gene, entire genes, and then we also have base-by-base coverage data. That isn't something I'll talk about today, but that's another valuable kind of data that you can mine to try to find things that have nothing to do with annotation, right? So, and maybe not even anything to do with splicing. And so very quickly, and I'll do this one particularly quickly because there's already a lot of great material walking through how to use this package in its vignette and also on its website. But this is a very quick view of one way that you can use recount. So again, I'm loading a few libraries. I'm downloading a study, and in particular, I'm downloading the gene the gene level summary for this study. So this now is going to be a matrix where the rows are genes. If you want to, you can go check out the metadata for that study by sort of punting out to the SRA and seeing its metadata page for it. That can be useful because the abstract is always an incredibly helpful thing to read if you want to know what they actually did and what the annotated metadata actually means. Back to our... in this example, what we do is simply gather some of that metadata and use it to distinguish between the different groups in this particular study so that we can then do a differential expression analysis. And once you've done, and this is using DE-seq, this is not, this is a standard technique that folks use to do differential expression, and you can make plots and things like that. So this is this is an MA plot from the DE-seq results. So like I said, I went through that one particularly quickly because there's lots of good material for walking through exactly how to use the various functions in this recount package. So if you wanted to analyze data at the level of studies, this is a very natural way to do it. And you can see that a lot of care has been taken to make sure that the metadata is, first of all, available, and then second of all, that there are even ways of taking that metadata and linking it with other databases. In this case, we link it with the metadata in Geo, a separate database, to try to augment what's already there, since metadata wrangling can be the key to being able to do a good reanalysis of a study. So the third and final level that I want to show of analysis is this topmost level, and I use the analogy of a telescope, right? So if what we're trying to do is make it easier for people to query and make sense of public data, the parts of the telescope that I just showed you are like kind of like the fat part and the middle part, right? They're good at collecting lots of light, but they're very hard at creating, they're not so good at creating focused pictures that you can actually look at. We need this sort of like skinny end of the telescope too, right? So everything that I showed you involves downloading summaries. But we could, instead of forcing you to download the summary and then work with it, which in some cases is going to require that you download gigabytes of data. That first exercise I walked through, I pre-downloaded a file that was about 17 gigabytes, which is a lot to wait for, but then again, you get this enormous matrix that you can manipulate the entirety of. But what most people want to do is they have a pretty focused question. They have a gene of interest, or they have a splicing pattern of interest. And they really just want to get back an answer. They don't want to download raw data first. So the topmost layer, this thing called Snaptron, puts some indexing on top of the summary so that it's much easier to answer a targeted question and get back just the answer instead of getting back a lot of raw data and having to process it to get the answer. Inside Snaptron, there's a collection of indexes that are targeted at different kinds of data. Some of the data in this database has the nature of being an interval on the genome, like an intron. Some of the data in this database has the nature of just being arrays of floating point numbers, like gene expression values. Some of the data has the nature of being text, because it's metadata, like the abstract of the paper from which the data was derived. So we have different kinds of indexing techniques for these different kinds of underlying data. And when people pose questions, we can take their question and split it up into pieces. You know, if they wanted to ask about, oh, I'm interested in this gene, I only care if a splice junction has at least this much coverage, and I'm only interested in liver samples, right? So that's got 3 different aspects to it. We can divide it up and deploy the different aspects to the appropriate indexes and compose the results back. And to show you one quick example, I'll go to my third tab here. So here's one where I actually don't have to do any prep. So I haven't pre-run any of these cells. There is one we're going to have to wait for about a minute for it to finish. So I'll get that going. There it goes. Switch back. So some examples of some questions that you can ask with this interface are, for example, let's say you have a favorite gene. Let's say it's ABCD3, which I'll actually show you another picture of in a moment. You want to know, for example, how many reads support the junctions in that gene across all these samples that we've analyzed in public data. Like, let's say one of those splicing patterns seems to be a marker for some condition that you're interested in. Wouldn't it be nice to know how specific is it to that marker? Or how prevalent is it, period, right? Maybe you think it's rare. Is it really rare? Well, let's ask. Let's actually see how many times it occurs in public data. You can ask about the tissue specificity of a junction, because one of the data sets that we have indexed is a study called GTEx, where a lot of different tissues from humans were assayed. In GTEx, asking how prevalent a particular splicing pattern is across samples is kind of like asking how tissue specific is it, because you're asking across several tissues. You can very easily apply a test to figure out is it tissue specific or not. Let's say you have two splicing patterns that may or may not be mutually exclusive, but they, for example, it could be inclusion or exclusion of an exon, of a cassette exon in the middle of a gene. And you just want to know for which samples is the fraction of pattern A versus pattern B high, or for which is it low. And you can order the samples according to the relative prevalence of two different patterns. Okay, having said that, let me go back to the cooking show. And it's done. Okay, great. And so what I can show you in this case has to do with a story that we investigated from a study from Genentech. So there was a study at Genentech where they did some RNA sequencing, basically to see if they could find unannotated, novel, completely unknown before then exons that were nonetheless inside annotated genes. So they analyzed a particular data set, which was this thing called the Illumina Body Map 2.0 data set, and they identified several novel cassette exons, novel in the sense that they were not in any gene annotation, or at least not in a gene annotation that they looked at. They then went to try to validate this by doing more sequencing. They did more in-depth and longer read sequencing using a MiSeq instrument. They were able to validate a subset. And one example that they gave was from that gene I mentioned before, ABCD3, even though it's from transmembrane region 2. And they found an instance, a novel instance of an exon of a sort of mutually exclusive exon situation, two exons that seemed to be, you could switch and choose one or the other. So one of the things that we did is we took this list of putative novel cassette exons and just asked how prevalent are they in Snaptron in our database of 50,000 samples that we'd analyzed previously. And what the query that I just put through is telling us is basically how frequently we see evidence for those exons. In other words, by asking how often do we see splicing on either side of that exon in the same sample, right? And so that's what this table is telling us. These are all events that they picked out. These are all exons that they picked out in that paper. These are all counts for how often we see them in our database, basically, and how many different samples do we see that event. And you can see in many cases, we see it in thousands of samples, right? So the fact that they were able to validate many of these, most of them, with MiSeq sequencing, it's not surprising to us, right? Because we can look at a huge public compendium and we can see they're there. So it's a very easy way to quickly get an idea of how prevalent is something that putatively is novel, right? So it's novel in the sense of unannotated, but it's not novel in the sense of wasn't seen before. It certainly was seen before in many samples. And another way that we summarize this is with a box plot here, which is actually from the paper, showing that if you take their calls and stratify them according to whether they were validated or not, and then ask how prevalent were they, we can see the ones that validated were more prevalent in Snaptron. In other words, we kind of could have predicted which were the ones that were likely to validate, versus the ones that were not so likely to validate by looking at public data. And we look not just at that compendium that I just showed you, but also at GTEx, which is another great big compendium of expression data. Okay, so with that, I'm sure I'm a little over time, so I'll wrap up. I think one of the things that the future holds, not just for researchers, but for how we train, is trying to make the best use of public data. Because public data, I think, even though it's something that we value, right, it's important to us that it be available for reasons of the scientific method on top of reasons of being able to reuse it for other purposes. We certainly pay for that privilege because these archives take a fair amount of money to maintain. There was even a debate, which some people might remember around 2011, about whether to close the sequence read archive because it was too expensive. But given that we have this data, I think we should make it a regular part of what we do day to day and a regular part of how we train. Before we go collecting a huge expensive collection of samples, ask, is there really nothing that we could have done with what was in the archive? Because we could start to ask that question immediately, or we could start to figure out a better design for our experiment immediately. And this is a view that others have expressed as well. There's another important aspect here, which is that this isn't just a computational problem. So I don't want to stand here as a computer scientist and say, all we have to do is index it, because that's certainly not true. There's another set of problems that come with how do you even work with data that's from a collection of disparate studies. And statisticians are very familiar with this notion, right? As soon as you put together data from multiple studies, you find that the biggest variables that distinguish the data sets are not the biological variables of interest, they're confounding technical variables. So people just going by names like lab effect and batch effect and things like this. So with public data, what that's essentially allowing us to do is sort of travel to the right on the spectrum of where maybe day to day we're used to getting data sets from our collaborators and we analyze them one at a time. Then all the way over here is we go to the whole SRA and ask, how prevalent is this, or something, ask differential expression or something. Not every question is going to be one that we are really able to answer. We're sort of encountering more and more dragons as we go to the right along this line. And I think a very important matter that needs to be worked out, and it's clearly not just a computational problem, is what are the questions that we can ask? What are the questions we can ask and answer robustly as we go to the right? There's some work on, and another big important problem here is as soon as you pool data across many studies, you're subjecting yourself to more and more issues with metadata. You're at the whim of more and more people in terms of how they enter their metadata. A big question is how do you deal with wrong or missing metadata? And my colleagues have some work on how you can actually infer incorrect or missing metadata based on the data. But that's really just a starting point. Metadata standards for the SRA are not the best. There's certainly incorrect data in there. And it's going to be very important that we understand and properly wrangle metadata into the future. So not all the problems here are computational by any means. And so with that, I just want to conclude, and thanks to the folks who did all the work and to our wonderful funders, and happy to take questions. Yeah. Yeah. Yeah, I mean, one issue with the way we've done it is we are able to make statements like this about individual junctions, but we're not putting together isoforms. We're not assembling entire annotated isoforms. Whereas that's usually the form that annotations take, right? The annotations are telling you about all the isoforms of a gene. So on the one hand, no, but on the other hand, sometimes the splice junction data is what you're looking for in order to inform your analysis. Like, for example, sometimes spliced alignment tools will use knowledge of where splice junctions are to basically boost their ability to align reads across them. So there, you don't care about the isoforms per se, you really just care about the junctions. So yes, we can compile resources like that. And there's another result, which I'm not sure if I kept it in my deck, but it basically, yeah, here you go. Basically shows that if you look at our compendium and ask basically, what was the advent of each splice junction? In other words, what was the deposition date of the very first data set that included that splice junction? And then look at that cumulatively over time. What's interesting is it levels out. And in fact, it leveled out a while ago, like in 2014. In other words, we haven't really been discovering very many new junctions in human since 2014, even though there's been a huge amount of deposition of data since then. So another part of the answer to your question is, now's a good time, right? You know, this would not have been such a good time, but now we're in the level off period. So it might be a good time to assemble resources like that. Yeah. Mm-hmm. Oh, so what if we limited our attention to protein coding genes? That's a good question. We haven't done it. We haven't stratified it that way. So you're absolutely right that this will include, non-coding genes, which are also spliced sometimes, and it might also even include splicing where, if we really showed it to someone, no one would believe us, right? Because it's not anywhere near. But I don't think that's a good question. We haven't broken it down to just that are within the confines of an annotated protein coding gene. Yeah. And that, by the way, the annotation we're comparing to is gen code in this example. Any other questions? Yeah? Yeah. You're right. Yeah, indicating that maybe they should have validated. Um, right, so what's going on here, for example, right? You're pointing out that this one had a high count. In other words, we found lots of splice junctions on either side of that putative novel exon, indicating that we think it's there, but they themselves were not able to validate it in their second data set. We don't know what's going on there, but their second data set, you could say maybe that's the issue, or you could say perhaps there's something systematic about the errors that are being encountered, so that you know we consistently found these and we consistently found them in the same samples, but it's not real. I would tend to think that it's probably an issue with how they validate. Oh, by the way, that rightmost column, shouldn't worry too much about that all being false. I made a mistake in the analysis. Only some of those were even attempted. There's only 9 or so that were ever attempted in that final column. So false there doesn't mean it failed to validate. It just means my annotation was off. But that second to last column is, in fact, the validation status from their MySeq sequencing. I would guess that that's a missed validation, that it should have validated, but I don't know. Question? Yeah, more? Mm-hmm. Yeah. Yeah, so like one of the things, for example, that we enable people to do is if they have a splicing pattern of interest. So we did that here, for example. If they have a splicing pattern of interest and they want to see is it prevalent across tissues, right? They can either ask that, these are basically 2 different compilations that we've indexed. So people can ask that question over this compilation, which is all kinds of data sets from the sequence read archive, or specifically over GTEx, which is various human tissues. And in fact, it's many of the individuals contribute many tissues just from that one individual in the GTEx project. Um, so sometimes we use it like here just basically because it gives us kind of like a good coverage of cell types, right? So if you want to ask, does something occur at all, it's kind of nice to have that somewhat even coverage from different cell types. But sometimes you want to know, is there specificity to a particular tissue? And so we have a special, we have a particular, this second bullet here is that there's a very particular query where if you have a splicing pattern and you want to know, is it tissue specific in GTEx specifically? That's one of the questions that you can ask. Yeah. We try to enable people to do that. Yeah, yeah, absolutely. Top link. I don't want to rob you of any more coffee. Should we maybe